Hello everyone. Our today's topic is Funaria. Funaria is a member of bryophyte. This is also known as quad mouse. This presentation made by some students like you. It means you can also prepare and you win. Definitely you win. This presentation made by these 4 students: Gamit Anirudh, Akshay Pandya, Ashmira Pathan and Patel Kushi. I really appreciate their keen interest to complete this presentation. I am impressed with their ability to follow all instructions to prepare this presentation. So first, we start from the classification. But before this, I want to say something. This is my humble request to all. Please watch this presentation till end. These students did a very hard work and they added some important MCQs, multiple choice questions at the end of this presentation. So please stay connected with me. So let's start with classification of Funaria. Division Bryophyta, class Bryopsida, order Funariales, family Funariaceae, and genus is Funaria. So this is a brief classification of Funaria. Kingdom Plantae, multicellular eukaryotic autotrophic organisms that are primarily terrestrial, possess cell wall made up of cellulose, and have a unique mode of reproduction involving alternation of generation. Next is division Bryophyta. These are non-vascular plants that lack root, stem and leaves. They exhibit an alternation of generation with the gametophyte stage being dominant. So class is Bryopsida. The true mosses, most of the familiar mosses belong to this class. They have a well-developed protonema stage, which is the initial phase in the life cycle of mosses, resembling a filamentous alga. Order is Funariales. It contains moss species which have a similar growth pattern and reproductive structures. Family is Funariaceae, characterized by their pear-shaped capsule and the presence of peristome. Peristome is a structure surrounding the opening of the capsule and aids and help in the spore dispersal. Genus is Funaria. This genus includes moss species. One of the most common species within this genus is Funaria hygrometrica. So habit and habitat. Funaria is the most prominent genus in the family Funariaceae. It is represented by about 117 species and is found in all parts of the world. Funaria hygrometrica is the most common species and is called quad mouse due to its twisted seta. They have a characteristic sporophyte with a long seta and a pear-shaped capsule. When mature, the capsule opens to release the spores, which help in the reproduction and dispersal of the moss. The presence of the hygroscopic ring around the mouth of the capsule makes it sensitive to changes in humidity, causing the capsule to open and close and aiding in the dispersal of spores. Some of the common Funaria species are Funaria hygrometrica, flavicans, Apophyta, etc. So, external morphology. This part of the presentation will explore the morphology of Funaria, a genus of non-vascular mosses. Next one is introduction. Funaria is a genus of mosses, a group of non-vascular plants belonging to the bryophyte division. Funaria mosses are commonly found in various terrestrial habitats worldwide. First is gametophyte. The dominant and visible phase of Funaria life cycle is the gametophyte. It is a leafy photosynthetic structure that grows vertically and resembles a miniature tree. The sporophyte is the short-lived diploid phase in the life cycle of Funaria. It consists of capsule and calyptra. Gametophyte. Stem. The central cylinder stem or axis of the gametophyte provides support and conducts water and nutrients. Next is dendroids. These are hair-like structures that anchor the gametophyte to the substratum and help with absorption of water and minerals. Dendroids are not tree roots as they do not perform the same function. Funaria gametophytes have small, lens-shaped leaves arranged spirally around the stem. These leaves are typically a single layer of cells, thick and contain chloroplasts for photosynthesis. Sporophyte. So capsule, this is a small elongated structure at the top of the seta. It contains spore-producing cells. When mature, the capsule opens to release spores. Next is calyptra. You can see in the figure a protective hood-like structure that covers the immature capsule. It is derived from the archegonium and often remains attached after the capsule opens. Funaria anatomy. So, tiers of axis. Internally, there is no much specialization of cells. The axis or the stem is differentiated into three different regions: epidermis, cortex, conducting strand or medulla. Stem is circular in outline. Single-layered epidermis is the outermost layer and consists of small chlorophyll-bearing green cells, but mature stem lack chlorophyll. Below the epidermis, there is a cortex present. It consists of large, thin-walled parenchymatous cells. At the maturity, cortex is observed to be differentiated into outer thick-walled, inner thin-walled cortex. It may also contain chloroplasts. So, innermost region of conducting strand or medulla consists of narrow, thin-walled and vertically elongated cells. Its function is to provide mechanical support and help in conduction. There is no xylem or phloem. Next one is tiers of leaf. Leaf consists of central midrib and wings of either side. There are no mesophyll or stomata in leaf. Midrib is several cells thick and contains many thin-walled cells of conducting strand. Thin-walled cells are covered by outer sheath of thick-walled cells. The cells of the wings are large green parenchymatous and contain many chloroplasts. Sex organs. Sexual reproduction is bogamous-type and takes place by Antheridia and Archegonia. Funaria hygrometrica is monoecious and autoecious. Archegonia and Antheridia develop on two different branches of the same plant. On the species of the male and female branches, develop many Antheridia and Archegonia, respectively. They are surrounded by pericytian leaves. Antheridia, they are present at the tip of the male branches. Antheridia remain surrounded by many pericytian leaves. Many long green sterile filaments also develop along with Antheridia. These are known as paraphyses. Each paraphysis consists of 4 to 6 cells with an upper large globular cell. Many Antheridia in different stages of development are present on male branch. Each Antheridium consists of a massive stalk and a club-shaped body. You can see this in the diagram. A sterile layer of jacket is present outside each Antheridium, the apex of which is closed by 1 or 2 opercular cells. Jacket is green when young but becomes dark at maturity. Jacket encloses many endocyte mother cells, each of which into 2 endocytes. each androcyte metamorphosize and convert into biflagellate antherozoid. Next is archegonia. These are present at the apex of female branches, surrounded by pericytales. Along with archegonia, many hair-like long multicellular paraphyses are present on each female branches. A mature archegonium is a flask-shaped structure with an enlarged venter and a long neck. Venter remains surrounded by a double-layered wall. You can see in the figure. Neck consists of 6 vertical rows of cells and encloses 6 to 8 neck canal cells, a ventral canal cell, and an egg cell. Next is sporophyte. It is divided into foot, seta, and capsule. Foot is conical in shape and remains buried in the upper tissue of the gametophore. Its functions are absorption and anchorage. Seta is very long, cord-like, reddish-brown structure, and it's held the capsule high. Internally, seta is differentiated into thin-walled cells of conducting strand surrounded by many layers of thick-walled cortex and an outermost layer of epidermis. Seta is hygroscopic in nature. Capsule is a pear-shaped structure, the upper portion of which is covered by a gap-like calyptra, which later on falls off. Capsule is erect when young, but become pendant at the maturity. Capsule is differentiated into apophysis, theca, and operculum. Apophysis is the basal, green photosynthetic region of the capsule. Internally, it is differentiated into central columella, surrounded by mesophyll and outermost epidermal layer. Theca is the middle, spore-producing region of the capsule. The uppermost lid or cap-like region of the capsule is the operculum, which helps in the dehiscence of capsule and dispersal of the spores. So, operculum remains connected with the capsule by the annulus. You can see in the diagram. On the left side, you can see apophysis, and right side, this is the LS of capsule of Funaria. Next one is theca. Capsule wall in the region is bounded by a layer of epidermis with very few or no stomata. Below the epidermis area, very few layer of colorless parenchymatous hypodermis, which surround by 1 or 2 layer of green chlorophyll-bearing spongy cells. Below the spongy layer, there is a region of air cavities. By many trabeculae, it surrounds the spore sac. Spore sac is filled with the spore formed by the reduction division in the spore mother cell. Inert to the spore sac and just in the center of theca region is present a solid column of sterile tissue called columella. This slide is very important, so read very carefully. C is operculum. It is a circular cup-shaped lid present at the top of the capsule. At the base of operculum, there is a ring of epidermal cells called annulus. So, annulus is present. Next one is peristome. If cap-like operculum is removed from the capsule, many tooth-like projections are seen at the top of the capsule. These are known as peristome teeth. Teeth in the peristome are long, conical, and present in 1 or 2 rows. In each row, there are present 16 teeth. They are hygroscopic in nature. They help in spore dispersal on opening out in the dry condition. So, till now, we learn so many things about Funaria. So, let's start revision of this chapter. Start from vegetative reproduction. It takes place by primary protonema, secondary protonema. So, difference between these two protonema are Primary means the spore germinate to produce branched, filamentous and multicellular structure called primary protonema. These cells break into fragments to produce new protonemal cells which develop into a leafy gametophore. By secondary means a protonema that is developed from any other means than the germination of a spore. This is called secondary protonema. It is developed from broken or injured rhizoids, stems or leaves. It can give rise to a whole new gametophore. By bulbils. Bulbils are small resting buds that arise from the rhizoids. They start dividing in favorable conditions and produce filamentous protonema that can grow into leafy gametophytes. By gamy. In unfavorable conditions, the terminal cells of the protonema divide transversely and longitudinally to give rise to 10 to 30 celled green bodies. These are called gamy. These structures start to divide and form new plants in favorable conditions. Apospory. Apospory is the development of gametophyte from sporophyte without spore formation. Vegetative cells of the sporophyte give rise to green protonemal fragments which later become a gametophyte. The gametophyte produced from apospory is diploid that gives rise to tetraploid and sterile sporophytes. Sexual reproduction. In Funaria, sexual reproduction is of oogamous type. It means two types of sex organs are present. The male reproductive structure is antheridium and the female reproductive structure is archegonium. The plant is monoecious. Monoecious means both male and female reproductive organs on the same plant. And autoecious. Autoecious means male and female reproductive organs grow on different branches of the same plant. The main shoot of the gametophyte bears the antheridium in clusters and acts as the male branch. The female branches arise as lateral outgrowth from the base of the male branches and bears archegonia. The female branch grows higher than the male branch. Funaria is protandrous, meaning the anther matures before the archegonia to promote cross-pollination. Antheridium. This is the male sex organ. The antheridial branch is about 1 cm in height. The branch bears reddish brown to orange antheridium in different stages of development. The antheridia are surrounded by a rosette of leaves called perigonial leaves. The antheridial cluster along with the leaf rosette is known as the perigonia. Numerous club-shaped sterile and hair-like structures are found in the antheridia that are known as paraphyses. They store water, help in photosynthesis of dairy cells of antheridia, and also protect the growing male reproductive organs. You can see the structure of antheridia on your right side. The antheridium is a club-shaped structure that has a short and stout multicellular stalk. The antheridial body is massive and elongated. It is covered by a layer of sterile cells called antheridial wall or jacket. The apex of the antheridium has a few layered thick wall called opercular cells. The androcyte mother cells are present below the wall layer. The androcyte mother cells divide to form two androcytes, which further gives rise to biflagellated and elongated sperm or spermatozoid. At maturity, the opercular cells absorb water and swell up. The walls of the opercular cells and androcytes get gelatinized, and a terminal pore is developed at the tip of the antheridium. The androcytes are released from this pore covered in mucilage. The mucilage is dissolved in water and androcytes develop into biflagellated antherozoids. Next one is archegonium. This is the female sex organ. The female branch is born higher than the male branch. Archegonium is born in clusters at the tip of the archegonial branch. The archegonium is a flask-shaped structure It consists of a neck, a swollen venter, and a narrow neck. It is attached to the female branch by a stalk. The venter consists of a venter cavity surrounded by a double-layered venter wall. The venter cavity contains a ventral canal cell and an egg cell. The neck consists of an axial row of 8 to 12 neck canal cells. The tip of the neck is closed by 4 cover cells. At maturity, the neck canal cells and venter canal cells disorganize to form a mucilage. The cover cell of the archegonium separate, forming an open passage leading to the canal. Next is fertilization. The spermatozoids or antherozoids reach the archegonium by pollination. The mucilage present at the mouth of the archegonia attracts the spermatozoids towards them because malic acid is present in this mucilage. The spermatozoids then enter the venter cavity through the neck canal. Then one antherozoid fuses with the egg cell and form a zygote, which later develops into a sporophyte. Now, you have to solve yourself these MCQs. Question number 1. Sensor mechanism of spore dispersal is seen in. Next is in Funaria, the peristomial teeth are. Next one. The reason why Funaria is a bryophyte is. Before seeing the answer, next in Funaria, the sporophytic phase is well developed, comprising rupturing of dash causes the dehiscence of moss capsule. Try to give answer yourself. From protonema, leafy gametophyte is formed in Funaria. The antherozoids are the spore producing organ of bryophyte is. Very good. Gametophytic generation is dominant in. Peristome teeth are present in. Please give answer by yourself. Don't see the answers. Sporophyte of Funaria is amphibians of plant kingdom. Right. Protonema is which is not a part of moss capsule. Anchorage organ of Funaria are correct. The dehiscence of moss capsule takes place by rupture of Funaria gametophyte is. The peristome of Funaria has. Thank you very much. We will meet in our next lecture. Hello.